Hello, Natasha, how are you?
I'm good. How are you?
Good.
I want him to take it
You want him to take out your baby.
right there, but he did not.
He didn't tape it? Where'd you want him to take it to? Okay.
But he did
How is he
not.
doing?
He was doing good yesterday, but he woke up this morning in pain. So
Oh,
we had to give him pain medicine.
oh,
Hopefully once it kicks in, I can try to get him to eat some, but
You. That's me.
good job.
No, that wasn't.
The tape's not here. Go ask Daddy in the office if he has the tape.
So can you ask him?
Well, Mommy's getting ready to get into a call for school. Can you go ask Daddy if he has the tape? Right. I love you.
When you get to school, can you do it?
Absolutely. You want to say hi, Dr. Reddy?
hello.
Okay. Ask Daddy, okay?
No, when you done with that,
Okay.
can you go?
Yeah, give me 1 second. I need you to hop off. Mommy's gonna take notes. Sorry.
Natasha, did you see another paper recently published
Mom,
No, I did
saying
not.
that Alzheimer's is connected to periodontitis?
Really?
Yeah,
Who published? What journal was it published in?
I sent to Dr. Hankins. I can see.
I have to go. Eh, your house there.
It is published, I think I need to check the journal name, whom they share with, but recently I have seen. You can say Alzheimer's and periodontics and Google, probably you will get there.
Sweet. I'm definitely gonna have to look into that.
Yeah.
Ow. Mama.
So hopefully a few more students join.
Hopefully. I know I usually am scheduled to teach on Sundays, but
I
where
see.
Jace had surgery, we're home recovering.
Okay.
So I got my substitute and gave her the lesson plan for today.
Hmm. You work with a dentist and also you teach? Hmm. That's a lot of work.
Little bit, but I only teach on Sundays, so it's not too bad. I was teaching Sunday morning, Sunday night, and Wednesday nights, and I just had to cut back.
Okay.
I have many irons and many fires. Plus, I
Hmm.
help run my husband's company.
So your husband has a company?
Mhm.
What company is that?
I'm sorry.
What company is that?
He owns Gideon Consulting, which is a subcontractor for DataView. So he does IT and data consulting with government contracts for the state of West Virginia, Alabama, and California.
Oh.
Sorry. And then he also on the side he is a tech support for local dental offices, dermatology, doctor's offices. And then he's also helping with a new greenhouse that's trying to get up and running in Buffalo. So
Oh, wow.
trying to get their greenhouse and
Hmm.
hydroponics and all that stuff set up.
I see.
So,
Wow. I would like to see that facility someday.
yeah, once they get all
Yeah.
the red tape and bureaucratic stuff jumped through, I'm sure they'd be more than happy to give you a tour.
Great. Yeah, I'm also interested to start something like that. When I
Oh, yeah?
retire, I want to start something hydroponics.
Ooh. See, I don't know much about hydroponics, but my husband, if it involves a computer, he can figure it out like that. I envy him. I am not a computer person at all. There's a reason I married a tech guy, so I don't have to do it.
Well, you both are busy people.
What's wrong? What's wrong? What's wrong? What's wrong? Are you in pain? What do you want? Okay, okay. Shh. So let me.
Okay, so you open the question answer. First question in the study guide. Do you all have study guide opened? So please open the study guide or print and hold it in hand. So the first question is, narrow 3 classical experiments that prove DNA is the genetic material and heritable. So the first experiment, this is all in page number, in the book, it is page number 3, page number 4, and page number 5. 3, 4, 5, page number 5 and 6, 3 to 6. So what is expected? That's a lot of pages. You don't have to write like that. So it's a 10 points question. So what you guys need to write is first Griffith's experiment. that is Streptococcus pneumoniae, smooth cells, is the one that causes pneumonia, and rough cells is the one that cannot cause pneumonia. Streptococcus pneumoniae, rough cells cannot cause the pneumonia. The reason is the protein material around the cell wall of bacteria of the Streptococcus pneumonia's rough cells, resistant proteins of host can easily bind and inactivate. Whereas Streptococcus pneumoniae smooth cells that slips away from such resistant proteins, that's why it infects pneumonia. But you don't have to write this background also. You just write Griffith conducted an experiment, first experiment, that is heat inactivated virulent smooth cells. Okay, first point is Griffith heat inactivated virulent smooth cells and injected into the, mixed with the rough cells. So the rough cells eventually got converted into smooth cells. So that makes to understand that something from the smooth cells entered into the rough cells. So that is the first experiment, which is called transformation factor. So in 10 points, that is for 3 points. And next in 1944, 3 scientists, Avery, McLeod, and McCarthy, what did they do? They wanted to understand what is the transformation factor. So they used 3 different enzymes, DNase, RNase, and protease. DNase deactivates DNA, protease deactivates protein, RNA deactivates RNase, deactivates RNA. So one thing at a time, they took the smooth cells extract, heat inactivated it, that extract, but added to that 3 aliquots, made it in 3 aliquots. First alicot, they added protease so that the protein gets degraded and RNA and DNA are intact. For the second alicot, they added RNase so that RNA is degraded and protein and DNA is intact. And third one, they added DNase so that DNA is degraded and protein and RNA is intact. When they are all mixed with smooth cells, rough cells, and injected into the mouse, the first one, mouse did not die because the DNA was intact, only the RNA and DNA was intact, only protein is degraded. In second alicot, where they used RNase treatment, RNase degraded RNA, but kept protein and DNA intact. That's why that still extract could convert rough cells into smooth cells, and when injected into the rats, rat survived. Rat died, sorry, because the DNA is the transformation factor. And third one clearly proved because the third alicot, when the DNase was given, the treatment to that alicot, DNase deactivated DNase deactivated DNA, but kept RNA and protein intact. But that since the DNA is the genetic material, that transmitted rough cells to become smooth cells. When injected into the rat died. So that is the second experiment. And third experiment is Hershey and Chase. What kind of experiment Hershey and Chase did? They used bacteriophage. So again, sorry, I did not use PowerPoint. So the first experiment is, it is not mentioned here, 1928, that is Griffith's experiment, where inactivated smooth cells mixed with rough cells, that converted rough cells into smooth cells. And second one is Avery, McLeod, and McCarthy. This is the experiment where the smooth cells extract is here, inactivated smooth cells extract. Inactivated or activated, you don't have to inactivate it. RNase is added that destroys RNA, protease is added that destroys protein, DNA is added that destroys DNA. And when the cultured with rough cells, smooth cells appeared in this case where RNA is degraded, protein and DNA is intact. So these smooth cells are showing up here. And here also smooth cells are showing up because protease degrades protein and RNA, protein, but RNA and DNA are intact, so smooth cells are showing up. Whereas DNA destroys DNA, so that destroys DNA, so the DNA is the genetic material, so none of the smooth cells none of the rough cells showed any smooth cells, smooth colonies, indicating since the DNA destroys DNA, so once the DNA is not there, it is not transforming. So this clearly shows that the DNA is the genetic material, not protease and RNAs. And the third one is Hershey Chase experiment, 1952, where you have this bacteriophages, which can be labeled differentially. That is the bacterial capsid, the bacteriophage outer coat is protein, capsid. Capsid is labeled with S35 and DNA is labeled with P32. You see, P32 is labeled DNA shining, whereas here coat is shining. So when such a virus or bacteriophage is infected to the bacteria, so the bacteria produces bunch of bacteriophages, daughter cells transmitted from this, but all of them had their DNA labeled. This was showing up in the progeny bacteriophages, indicating that the virus has, this bacteriophage or virus injects DNA, not the protein. The protein, the coat remains outside, only it injects the DNA. You see tiny thread there. If the DNA is labeled, the progeny's DNA is also labeled with P32. So these three experiments clearly show the DNA is the genetic material and also heritable here from parental bacteriophages to progeny bacteriophages. So that is the first question. You can write it in a small paragraph. Like 1928, Griffith inactivated smooth cells and mixed with rough cells and transformed some of the rough cells into smooth cells, number one. Number two is Avery, McLeod, and McCarthy did RNase, protease, and DNase treatments to the smooth cell extracts and mixed with the rough cells that kept only RNase protease experiments were intact, so they converted into smooth cells, but not the rough cells, not the DNase treatment. So it is very easy. And third one is bacteriophage, which is labeled with S35 and P32. Transmission clearly could be traced in the progeny that the DNA is the genetic material as P32 label showed up in the progeny. So that's all. So you can write it in a very concise way. Don't have to draw a whole lot of it, and a whole lot of description is not needed. So the second question is about the Beadle and Tatum. So this is the experiment conducted by Beadle and Tatum. What did they do? They used this Neurospora crassa. Neurospora crassa is the red fungus that grows on the bread. Okay, so how it can be maintained? It can be maintained in the complete medium, in the minimal medium. So in the minimal medium or complete medium in the sense that has all things like secondary metabolites, amino acids, or nucleic acid precursors, vitamins, all nutrients are here. That's why it is complete medium and it is minimal medium. So Neurospora crassa spores, conidial spores, can be inoculated here. Inoculation shows here lush growth even in the minimal medium because Neurospora crassa maintains everything, can synthesize everything. It can synthesize its own vitamins, its own amino acids, its own secondary metabolites or nucleic acid precursors. Okay, but this one, the color is different because these asexual spores are mutated, X-ray irradiated. So Neurospora that is X-ray irradiated produces mutant spores. Mutant spores can be maintained in the complete medium. Why? Because complete medium has all the nutrients, but the mutants cannot be maintained in the minimal medium because the mutations disrupted some functional genes. Okay, so some functional genes related to amino acids, vitamins, or nucleic acid precursors are kind of dysfunctional. They cannot be synthesized by itself. So if you are just giving a minimal media, that won't help. But if you are giving them complete media, they can get complemented. So for example, if the mutations are in the vitamin genes, mutations are not in the vitamin genes, you add all the vitamins, but growth is not complemented. You add all the nucleic acid precursors, growth is not complemented. But if you added in the minimal media amino acids, growth is complemented. That clearly tells us there is mutation in the amino acid specific genes. So which amino acid? There are 20 different amino acids. So separately, minimal media is added with each different minimal media, each different amino acid. Minimal media plus leucine, minimal media for valine, plus glycine, plus serine, plus arginine, plus lysine, plus prolin. So now the growth is complemented when arginine is added. That means this mutation in this mutant Neurospora is in the arginine biosynthetic pathway. That's why you added arginine, that is doing well. So this is mutant screen. And then the arginine, fourth step is arginine has 3 different amino acid in the pathway: ornithine, citrulline, and arginine. Okay, so if ornithine is supplemented, it is not growing. That means ornithine is fine. Ornithine is the top. And second metabolite in the pathway is citrulline. If the citrulline is added, that is showing up. That means there is defect in the citrulline. So using this mutant screen, now we have identified that this mutant has defect in the citrulline synthesizing gene. That's why that's why as soon as you added arginine, since the final product is arginine, even at the level of citrulline, it complemented. Complemented in the sense it is growing well. So you can write it in your own language if you want to. You can say first Beadle and Tatum identified mutant screen, that is the normal Neurospora crassa can be grown in the minimal media and complete media, whereas the mutant that is X-ray irradiated, we do not know where it is got mutation, but in the minimal media it cannot grow, but in complete media everything can be grown. To find out where the mutation actually happened, minimal media is added with vitamins, amino acids, and nucleic acid precursors separately. And then when the minimal media with amino acid complemented the growth, that tells us that the mutation happened in these amino acid genes. To identify which amino acid precisely, because there are 20 different amino acids, Beadle and Tatum added different amino acids individually. That showed when the minimal media with arginine again gets complemented. And here, to understand, because in the arginine biosynthetic pathway, there are 3 different genes, ornithine, citrulline, and arginine. to understand where actually the mutation happened, they separately or individually mixed with the minimal media, ornithine, citrulline, and arginine, and that clearly complemented at citrulline, indicating that the mutation in this mutant spores is at citrulline. Either you give citrulline or the final product arginine that gets complemented. So this is for the second one, mutation screen. This is what Beadle and Tatum did. So when you are writing this 10 points question, first you need to explain complete media, minimal media, X-ray irradiated mutants, and why it cannot get complemented in the mutant media because we do not know where the mutation has happened. And then we can add at broadly amino acids or vitamins and nucleic acid separately, and then individually, and then each gene separately to understand where exactly the mutation happened. So that is the 10 points question. So then the complementation test is this one. So what is complementation test? Why it, what for we are doing complementation test? Because we have identified here two mutants, mutant A and mutant B. Now the question is whether the mutation occurred in the same gene or different gene to produce this phenotype. Phenotype is there is no synthesis. This one mutant A is mutant because this gene is dysfunctional. Once the gene is dysfunctional, in the figure it is showed like a, it is not folded well. Where this one is folded well, that means this blue gene is, this purple gene is intact. Red gene is mutant. But in this one, this mutant B also do not have the same phenotype. These do not have the same phenotype because red gene is all right, but blue gene is mutated. Both genes are needed for expression of normal wild type. Okay, so when they are mated, these two mutants, this gives a heterokaryon. Heterokaryon in the sense, just like animals or plants, fungus will not produce a zygote. It will produce filaments with individual nuclei in it. You have the blue nuclei and red nuclei coming from both sources. So red is coming from here, normal red, and the blue is normal red is blue is, I mean, this is blue, not purple, sorry. Blue is coming here. So at least two functional copies are there. This has, this nucleus is good for blue gene. This nucleus is good for red gene. So these two genes complement, and then there is a growth here. So this is the complementation test. But if the mutation is in the same mutant, like mutant 3, so what is problem with mutant 3? Mutant 2 also blue gene is defective, and then mutant 3 also blue gene is defective in different place. See here, mutation is here, mutation is here, but do not matter. When you cross it and make heterokaryon, what is happening? The red genes are intact because both red are good, but there is no functional blue gene. Okay, since both blue and red both are needed for complementation or growth, this is not complemented because blue is not there. Functional blue is not there. So this complementation test helps us to understand if the mutation is in the same gene or in different gene. So that is the question number 3. So that's all in this chapter. So we will go to the next chapter. So this is the next chapter where we have some questions. So one is comparing the monohybrid and dihybrid and how those led to the Mendel's discovery of inheritance, loss of inheritance. So from here we can discuss what is monohybrid is taking a single trait at a time. So the round, which is homozygous for round, and the recessive, which is homozygous for wrinkled. Okay, so they produce gametes, capital W and small w from this parent. And in heterozygous state, round is masking the effect of wrinkled. So the round is the dominant and wrinkled is the recessive. So then in segregation, in the female gametes are also capital W and small w, and male gametes also capital W, small w. So that gives like one homozygous capital W in the Punnett square, and one small w and one capital W, that is heterozygous, and then another capital W and small w between these two gametes. And then this outcome shows both the recessive alleles are combined, small w, small w. So we need to narrate this monohybrid cross and say one thing. What did Mendel identified here? Law of segregation. That is, the alleles are transmitted through the gametes, and then the gametes in combination will tell us that which one is heterozygous allele, which one is dominant allele, which one is recessive allele. And then in the Punnett square, we can see all the outcomes, homozygous and heterozygous and the homozygous recessive. That tells us this is the law of segregation. So the alleles come together and segregate. That is law of segregation. Okay. Whereas principle of segregation or law of segregation. Whereas in heterozygous, in the dihybrid cross, we are taking two things at a time. What are the two things? Color and shape. What are the colors? Yellow color, which is dominant, and green color, which is recessive. And independently, they segregate 3:1. And also seed shape, which is round shape and wrinkled shape. Okay. And in monohybrid cross, they segregate 3:1. Okay. So when we combine both of these, round and yellow gives 9 out of 16. Because 3/4 and 3/4, multiply 3/4 with 3/4, that is 9 out of 16. And then round and green are mated. 3 out of 16 are round and green. And here it is wrinkled and yellow are mated. 3 out of 16 are wrinkled and yellow. And then wrinkled and green, both recessive types, are 1 out of 16. So what is this telling? This is second law of Mendel, or principle of Mendel. What is this law? Traits, whether it is the seed color and seed shape, they are independently assorting. Okay. And when the alleles come together, we will see the phenotype. So here is neither color is influencing the shape, nor shape is influencing the color. They are all independently assorting. That is what we need to write. Since they are independently assorted, we have all combinations, parental types that are clearly yellow and green types, are round and wrinkle types, and then the combinations also. that is round and green type, which is a recombinant, wrinkled and yellow, which is a recombinant. These are the parental types, these are the recombinants. So we are seeing all of these segregants in the independent assortment. So this is what is the fourth question. So first to start with, from here, that the first law is law of segregation. We need to explain what is law of segregation. That is monohybrid, where the alleles in homozygous state produce gametes, and in gamete there will be one allele, and then in combination the heterozygous F1 will indicate which is dominant and recessive, and whereas in F2 you will find the segregation, which is outcome of combinations of various alleles. Whereas here in the dihybrid cross, you have seed color, seed shape. Here shape is round and wrinkled, color is yellow and green. And then you can see all the outcomes in various combinations. Like in monohybrid cross, yellow and green segregate 3:1, and round and wrinkled segregate 3:1. So 3/4 and 3/4 are 9 out of 16, and then 1 out of 16 are the parental types. Whereas recombinants are 3 out of 16. The recombinants are round and green combined. Unlike parents, the progeny has contrasting phenotypes from the parents. And here it is wrinkled and yellow is combined. So that is what we are going to say. Independent segregation. And then the next is explain the molecular basis of round and wrinkled seed shapes in peas. Why are molecular traits considered as codominant? So that one is this one. So where is that one? Yeah. So first you will start this question number 6. You will say that here the starch branching enzyme 1 gene is very important for producing amylopectin. Okay. So here is a transposable element that is present in the P genome, which is transposed right in the middle of the gene. So this mutant with starch branching enzyme 1 with the transposable element is associated with the wrinkled seeds because it cannot produce functional amylopectin. Whereas if this one, there is no transposable element, so this is associated with the round seed because it can produce intact amylopectin. Okay. And then why are these traits considered as codominant? Why it is considered as codominant? Here are the slots in the electrophoresis. We can do the PCR for this gene. So the first thing we need to explain is we need to do PCR for this gene and use those primers for amplification of round, wrinkled, and the F1. F1 is a cross between round and wrinkled. So here you will find different phenotypes. The first one has shorter band. This band is shorter. And then this is, heavier band. And this is lighter band. Lighter band comes from the wild allele because these do not have transposable elements, hence size is smaller. So in the electrophoresis, it can migrate faster. Whereas this one, it cannot migrate faster because it has transposable element. So this is heavier than this. Extremes are the parents. Round parent, wrinkled parent. Whereas this one is the F1, will have both bands coming from the wrinkled parent as well as the round parent. Because anyway, chromosomes have the allele. One chromosome has this allele and its homologous chromosome has this allele. That's why you see both the bands. So you can just explain from this figure or simply this one. This figure also will tell. So here what is showing, you are seeing from here, because this slot is where it is loaded. So round plant is producing lighter band. Wrinkled parent is producing heavier band because it has transposable element in it. Whereas heterozygotes, it has capital W allele as well as small w allele. So lighter band is coming from capital W and heavier band is coming from small w. Okay. Why it is heavier? Because it has transposable element. So that is the explanation for chromosome, question number 6. Have an pedigree analysis for the eye color. So I will open the 3rd chapter. Is it okay? Is it helping for everyone? Please write your comments. So especially undergrads, is it good? Natasha, Brandy, Sam. Good, very good. Cyrus. All right, so chapter 3. So chapter 3, what is the question in chapter 3? Discuss chromatin, nucleosomes, and histones. Hope this figure is there. Yeah. So what is a nucleosome? Chromatin is, what is chromatin? Chromatin is DNA and protein, highly coiled samples. Stable complexes of DNA and protein are called chromatin. Okay. So in the chromosomes, DNA and then protein are kind of made a stable complex. So what is there in that protein? The one, a bead-like structure, which is called nucleosome, that has histones. So what are the different kinds of histones? H2A, H2B, H3, and H4, 4 histones. Okay. So these four histones, H2A, 2 copies, H2B, 2 copies, H3, 2 copies, H4, 2 copies. So nucleosome is an octamer of all these histones. All these histones form nucleosome, which is a core particle. Okay. So around the core particle, 55 base pairs of DNA is wrapped around. Okay. So this is just wrapped around, DNA is wrapped around this nucleosomes, and then, 55 base pairs linker it has. a DNA is wrapped around the nucleosomes, and then 55 base pair linker between two adjacent core particles or two adjacent nucleosomes. And then that linker DNA is stabilized by histone 1. So that's all you need to write for this question. So for question number 7, first thing you are going to write, chromatin is made out of DNA and protein, making stable complexes. And what is in it? Nucleosomes. And what are the nucleosomes? Made out of histones. And what are the histones? H2A, H2B, H3, H4. So nucleosome is an octamer consisting these 4 histones in duplication. In the sense, 2 histones, H2A together, 2 H2A molecules, 2 H2B molecules, 2 H3 molecules, 2 H4 molecules, making a nucleosome. So the DNA is wrapped around that nucleosomes, and then 2 adjacent nucleosomes are connected with 55 base pair DNA called linker DNA, which is stabilized by histone 1. That's all is the answer for question number 7. Question number 8 goes, just this one, question number 8. So explain the cell cycle regulation. So here, everything starts with gap 1, which is typically 10 hours, where synthesis occurs. The entire DNA is getting replicated here. So once the DNA gets replicated, then it enters, for DNA synthesis, all the enzymes, proteins that are needed for replication are getting built or populated. Once the cell synthesizes all the proteins, enzymes that are needed for DNA replication, which takes typically 10 hours, cell enters into S phase, where DNA replication occurs. So this is all for DNA replication. And once the DNA is replicated, that is 9 hours it takes. If it is human, all 23 chromosomes or 46 chromosomes. In the mitosis, 46 chromosomes. The DNA has to be built. So which is DNA synthesis. This happens through this process called DNA replication. Okay. And then again cell enters into gap 2, where a lot of synthesis is needed for enzymes and proteins that are actually needed in the mitosis. This takes roughly 4 hours. So actual mitosis, prophase, metaphase, anaphase, and telophase, cytokinesis, all that happens in 1 hour. So for all these processes, enzymes and proteins are built in the G2 phase. Okay, so this is called cell cycle regulation. So that is the question for question number 8. And then what is for question number 9? Yes, this is for question number 9. So question number 9, once the chromosomes are stained, okay, if you stain the chromosome or paint the chromosomes, you have 2 domains. That is densely stained centromeres, around centromere. This is densely stained. That is heterochromatin. So heterochromatin is here orange colored. Okay, so that is there in telomeres, where telomeres also are highly repetitive sequences that are complexly folded here. Okay, here are the telomeres. So either you draw or you can say upon painting the chromosomes, you can see orange colored heterochromatin and then blue colored euchromatin. Okay, blue-colored euchromatin is in between telomeres and centromere. Okay, right here, or in the other arm it is here. So then how this is related to genetic distance is, answer is another figure, which is in the linkage. Sorry, this is another chapter. This one, folks. So for question number 9, ignore previous this thing. Here also you have everything. Heterochromatin you have around centromeres. Telomeres also is there, heterochromatin. But euchromatin is the one that is functional genes. So if you make a genetic map is corroborating the euchromatin. You see, here is one locus, here is the one locus. That is within the boundaries of euchromatin. Okay, because there are no functional alleles here, very less genes, very few genes in heterochromatin, or there are no functional genes. So you don't have any genetic distance here. Okay, so genetic distance is estimated between the PR and CN, and that shows as if it's a very narrow gap in the genetic map. Whereas physical sequence is huge. Physical sequence, if you do DNA sequencing, you have from one end to the other end. That is physical map. So physical map corroborates throughout the length of the chromosome, but genetic map corroborates only the euchromatin, because all functional genes are in the euchromatin. That is what you are going to say. So very simple question. Once you paint the chromosomes, you can identify heterochromatin and euchromatin. Euchromatin have functional genes. Heterochromatin do not. So when you make genetic linkage map corroborates only euchromatin, as there are genetic loci there. Okay. So heterochromatin do not have any genetic loci. That's why that do not corroborate with the genetic map. But once you sequence, you have physical map from one end to the other. That corroborates both heterochromatin as well as euchromatin. So that's what we are going to write. Again, we are going back to the previous chapter, which is chromosome, which is chapter 3. Yeah. So question is eye color. And 10, explain why eye color in Drosophila doesn't segregate similarly in reciprocal matings. Here it is for this question. So what is it here? You have this cross parents. Oh, sorry. Yeah. The female is homozygous for red eyes color. 2 X chromosomes are there. Female is homozygous and male is recessive, and other is Y chromosome that do not have any allele. So in the reciprocal cross, you have heterozygous female and then red-eyed male. Chromosome is coming from here. So when you mate both of these, you have 3 red-eyed females, because the gametes are coming from here and here, heterozygous, and this one and this Y chromosome making red-eyed male, and this white and this one, this Y is making white-eyed male. So this is the segregation. Whereas in reciprocal cross, if you use the same thing, white-eyed female, here you used red-eyed female, but here you used white-eyed female. When crossed with red-eyed male, what happens? So one chromosome, red eyes, is coming from here. White chromosome is coming from here. So it is in heterozygous condition. This female is red eyes, and then this male is white eyes because it has eyes color. So, and then you see the segregation. You have 50% white eyes progeny. That is because this chromosome and this chromosome makes this female white eyes. See here, 3 red eyes and 1 white eye. And here it is 50-50 because this white chromosome makes it homozygous white eyes. And this is heterozygous for red color, which is coming from this chromosome and this chromosome. And then this chromosome and this chromosome produces white males. So unlike Mendel did reciprocal cross, when Morgan tried to validate in the flies, it did not happen. So what Morgan understood from this is that the genes are located on chromosomes, because clearly this Y chromosome, a short chromosome that do not have any gene, and the reciprocal crosses are different. And then he understood it. The X chromosomes are the ones that are having the genes. So the chromosomes are the ones that are having the genes. Since this is X-linked trait, because Y chromosome do not have any genes, the short chromosome, which was occurring only in the males, that's why he named it as Y. And then the segregation clearly shows how the Y and X corroborating the I phenotypes in reciprocal crosses differently. So you need to explain these two figures. Either you write it if you want. You can say in one cross he chose red eyes, homozygous, and white eyes, which produced the F1s heterozygous for red in both cases. Segregation was, the segregation was 3 red and 1 white in the F2. Whereas the second cross F1 itself, it is different. When he used white as female, homozygous white as female, and red eyes as heterozygous, the F1 itself is different, showing the white color. You see in the F1, both were red color, just like Mendel identified. But in reciprocal, in F1, there was segregation. Some of them were red and some of them were white. Because homozygous female had 2 chromosomes that are W. Okay, but here it is 50% red, 50% white, because there are more white chromosomes. So these two, one you have homozygous white female, and then white male, and then 2 red in heterozygous condition. So Mendel could identify the differences between the reciprocal cross, which led him to understand that the genes are on the chromosomes. The most significant achievement in this experiment is to identify the genes are on the chromosomes, right? So that is about it. And then copy number variation probably is there in the next chapter. Chapter 4. Chapter 4. chapter 5 then. Not there in the power. Okay. Let us a little bit is there copy number, but what I actually you guys wanted to write is copy number variation. Okay, let us write from here. Book, it is in the multiple places, but what we can write is this unequal crossover. That is the duplicate genes left and right must align left with left, right with right, but unequal crossover can happen. That is left aligning with the right and right aligning with the left or vice versa. Because these domains have same nucleotides. If the left aligns with this right, or this right aligns with this left. So homologous chromosomes are misaligned. And once they undergo crossover, as a result of that, some chromatids will get excessive number, 3 triplication, and some chromatids will get single copy. Especially in the color alleles, for example, some chromatids will have 3 green or 3 red. Okay. So because of unequal crossover between the red and green, creating copy number variation. So, and also you can write copy number variation also occurs in the saliva amylase, starch amylase. So starch amylase is the enzyme that degrades starch. Let me see in the book where it is exactly. In the book, page number 129. In page number 129 in the book, you see this, page number 129, figure 4.19, that is amylase copy number. So amylase copy number can vary among humans based on the subpopulations. Some populations that are descended from hunter-gatherers will have lesser copies because they don't eat much of the starch. And then some of the populations that are descended from the pastoralists are more like collectors, seed or fruit collectors, fruit eaters. So they will have more starch amylase genes. So the copy number can vary from 5 to 5.1 to 6.9. So plus or minus 1. So I would say can vary from 5 to 8. Some people will have 5 copies and some people will have 8 copies of starch amylase gene. Okay, so this is about copy number variation. First thing we need to write here is the basis for copy number variation is unequal crossover. So what is unequal crossover? The misaligning and contributing to the chromatids that have varied number of copies. This is the genetic basis, which resulted in the red and green pigment genes. That misalignment causes various copy numbers, either increase of green or red. So this misalignment creates deletion of sometimes green, and then multiple copies of green or red, which is variation in producing that opal pigments, green or red. That is about question number 11. Question number 12 is, how do we identify genes are located on the same chromosome or different chromosome in neurospora crosses? Neurospora crassa, where is this? It's in the previous chapter. Yeah. So here what we are going to explain, question number 12, how the genes are located on the same or different chromosomes. So we have in the tetrads, the ascus will form tetrads, 4 different haploid cells. So there, if you have parental ditypes, only parental types, or non-parental ditypes. Non-parental ditypes, there is a figure. So let me see in the book. In PowerPoint, there is no good figure. Folks, this is the figure that explains that. That is page number 133, 133, page number 133, figure 4.23. So in figure 4.23, what do we have? Is here is the basis for formation of parental ditypes that they are on separate chromosomes. Genes are on separate chromosomes. So AB combined and formed one cell. And then small b combined one cell, small b combined one cell. Because they are all on 2 different genes, so independently cells formed in the tetrad. Whereas non-parental ditype, in the sense all of them are non-parental types. Okay, that one is, this is the most important. That's why I gave this question. Here it is capital A, but this chromosome is small one. See, this is dark brown color, light brown color. Light brown color has recessive alleles. Dark brown has dominant alleles. But here, the arrangement on the metaphase is, the first is dark and then light. Whereas here, both dark, both light. So here is light and dark. So that means when this chromatid and this chromatid are kind of forming a cell that will have capital A and small b. And this will have capital A and small b. And this one also will have small a and capital B, and small a and capital B. So the parental ditypes and non-parental ditypes, the cells can form anyway. the ascus can form this type or this type, 50% chance. So when you count the ascus, what is an ascus, folks? What is an ascus? Ascospore is this one. This is the ascus. Inside there are the ascospores. Okay. So this is result of sexual reproduction in Neurospora crassa. So here, what happens if it is on different chromosomes, as you have seen? A allele and B allele are on different chromosomes. You have either formation of parental ditypes, where dark brown are aligned, or capital alleles are here, and all small alleles are here. So the formation of parental ditypes or parental, non-parental ditypes are because of this. Capital A and small b combined, or small a and small b combined. So this will create either parental ditypes or non-parental ditypes equal. So if you are seeing, if you are making a cross, what is that you want to do in this experiment? You want to measure the distance between A allele and B allele, right? That's why you mated these two, Neurospora crassa. So how do you, now it clearly says there are not on the same chromosome, they are on different chromosome. If they are on the same chromosome, then you have this if they are on the same chromosome here, if there are no crossover, you can find on the parental ditypes. The same page number 133, figure 4.23 will tell us if they are on the same chromosome. If they are on the different chromosome, figure 4.24 will tell us. So what is on the figure 4.24? Both genes are on the same chromosome. Now we need to identify the distance. How do we identify? One is, if there is no crossover, you have parental ditypes. Or if there are crossover, one chromatid is capital A, capital B, and here is the crossover. So this one will have capital A, the crossover happens, capital A and then small b, and here small a and capital B, because of this crossover. Brown color, the dark brown one is having capital A, light brown one is having small b. So capital A and small b are combined because of this crossover. Or small a and capital B are combined. Or small a, small b will be there. So this kind of ascus that has 50% parental kind, that is capital A, capital B, small a, small b, and then 50% recombinants, that is called tetratype. Tetratypes. So you need to count the tetratypes. So what is the question? That's how you will identify. So in short, what is the question? How do you identify if the genes are located on the same or different chromosomes in the Neurospora crassa? So the first point is, when you mate contrasting Neurospora crassa, if you are finding only parental ditypes and non-parental ditypes in 50%, in the sense 50% of the ascus are PD, which is parental ditype, 50% of the ascus are non-parental ditype. So if PD is equal to NPD, That means they are on different chromosomes. There is no linkage. Then how do you say that they are on the same chromosome? Then you will see some tetratype. Tetratype in the sense that they have undergone crossover, and then small a, capital B combined, and then capital B, capital A and small b combined, which produced tetratype. So all 4 genotypes are observed in the ascus. Then you can say, well, they are all located on the same chromosome. If the parental ditype is equal to non-parental ditype, they will say, we can say that they are on different chromosomes. If all 4 genotypes are observed, that is parent A kind, parent B kind, as well as recombinants, then we say it is tetratype. And then the chromosome, these genes are located on the same chromosome. And then how the linkage is estimated? Number of tetratypes to the total number of tetrads times 100. And because it is haploid, say half times number of TT, number of tetrads, times 100 gives us the distance between the genes like capital A and capital B, A locus and B locus. Any questions here? So it's very easy that you are making the question number 13. First, you will say, when you are mating to contrasting neurospora types with the purpose of mapping genes between, the distance between the genes, capital A and A locus and B locus, what are you going to do? You will mate contrasting types. That is, one parent has all capital alleles, one parent has all recessive alleles, right? So in the segregation, if you are seeing parental ditypes looking like exactly like parents and non-parental ditypes, which are completely 100% recombinants, that means these 2 genes are located on different genes. But if you are finding the tetratype ascus that has within that one ascus, you have ascospores which are parental kinds as well as recombinants. That must be because of recombination between between the 2 loci, and then they are located on the same chromosome. That's all you need to write. You understand what I said? Any questions? If not, we can move forward. So 14, question number 14 is, I think, chapter. Again, chapter 3. This is the answer for question number 14. So if you want to write also, you can write, or if you want to draw, you can draw. Writing is also very simple. That is, here is the DNA, which is 2 nanometers diameter. Once it is wrapped around this nucleosomes, this is called histone fiber, which is 11 nanometers in diameter. So the 11 nanometers histone fiber is condensed in chromatin fiber, which is 30 nanometers. So 30 nanometers chromatin fiber is further condensed in 300 nanometers coiled coil. So 300 nanometers coiled coil is further coiled into chromatin fiber is further coiled into coiled coil. So first is naked DNA, histone fiber, chromatin fiber, coiled chromatin fiber, coiled coil, and chromosomes, which is 1400 nanometers in diameter. Okay, so what if you want to write this question, you need to remember DNA, histone fiber, chromatin fiber, coiled chromatin fiber, coiled coil, and chromosomes, and then their dimensions. DNA is 2 nanometers, histone fiber is 11 nanometers, chromatin fiber is 30 nanometers, 300 nanometers is coiled chromatin, and coiled coil is 700 nanometers, and chromosomes are 1400 nanometers. You have to remember all this. Okay. So that is the answer for question number 14. And then cis and trans configurations are there in the next chapter. Question number 15. So what are the cis and trans configurations, folks? You know that. Not a big deal, but so what is cis? The mutant alleles of both genes are on the same chromosome. Here is mutant alleles. Capital A, capital B are wild alleles. They are cis configuration. And then trans is in the repulsion. That is one wild allele and one mutant allele. Mutant allele for B, small a and capital B. Capital alleles are the wild alleles. Small letters are mutant alleles. Okay, if both wild alleles are together, that means they are in cis. If the wild alleles are distributed on two different homologous chromosomes, that means that is trans. So how do we choose parents for designing a cross? Whether it is there here, it's not there here. So in the book, page number 111. Page number 111, and this is the figure. Either you can explain it and write it. So what are we going to write it? First of all, we wrote cis configuration. Isn't it? This is, yeah. What is cis and trans configuration in this figure? In class 1, you chose parents differently. That is white-eyed female. White-eyed female is because it has white eyes on both X chromosome, white alleles, small w, small w. And then why did we write plus plus? Because the wing is normal wing, whereas the male is red eyes with short wings. Okay, plus for red, that's why it is red eyes male and short wings. That is miniature wings. So you made it in the F1 in trans configuration, because you see plus is here for I, whereas for normal wing plus is here. So this is the trans configuration, whereas cis configuration is Only that part you need to write. We don't need to estimate any linkages. But here, you chose a totally recessive plant, a recessive fly, which is small w, small m, small w, small m. And here is the male is totally wild type, capital plus, capital plus. Now, this chromosomes comes here. Now this and this has the same phenotype, but orientation is, this is cis, this is trans. Can you do that? Page number 111, figure 4.2. For the question 15, question number 15. That is explain cis and trans configuration and choose parents and design crosses to construct F1s in cis and trans. F1 phenotypes are the same, but in one F1 it is cis and one F1 it is trans. So what you need to remember is, for trans, you need to select parents, especially the female parent, heterozygous. Okay? And then for constructing the cis, you need to select that female homozygous recessive. That's all. You need to know that. Okay. So question number 16. Yes, this is the question number 16. So explain the pattern of segregation of sequenced tagged tandem repeats in human genetic mapping, and how is it important in paternity test and forensics? So first we need to explain here STRs, DNA polymorphism. So what are the SSRs? SSR, the tandem repeats, where they're all over randomly. So these tandem repeats look like this, TGA6 or TGA8. So this, using this in human pedigrees, you can test like that. So here is a person having 4 repeats and 5 repeats, and sorry, female, and then a male is having 4 and 6 repeats. Okay, so the progeny of them has 4 repeats and 6 repeats. 6 is coming from dad, 4 is coming from mom. Alternatively, they could have had 5 and 4. Then we will say this 5 has come from female and 4 has come from the male, because dad doesn't have 5, dad has 6, but 4 is common between both parents. Whereas in this 1 and 2 repeats in the female, 3 and 4 repeats in the male. So this boy has 1 and 3. This girl has 4 and 6. Okay. Now they had, unlike their parents, they produced many babies. So now you can see 4 came from here, from mom, and 1 came from dad. This daughter, 1 came from dad and 6 came from mom. And here 3 came from dad, 4 came from mom. So either you drop to this and explain. So for this, what you need to write is, in human genetic mapping, you need to identify DNA polymorphisms, number 1. Number 2 is DNA typing identifies the number of repeats, if it is repeat lengths. Okay. So the repeat length alleles are diverse. Parents will have diverse alleles. For example, a mom had 4 and 5 and dad has 4 and 6. If their baby has 4 and 6, that means 4 came from mom and 6 came from dad. And then in another family, it is 1 and 2 and 3 and 4. producing a boy which is 1 and 3, because square is for man. And then the progeny siblings will have a diverse number of alleles, like 4 and 1, 6 and 3, 3 and 4, all kinds of combinations, 1 and 6, 3, 6, 3, 5, 1, 4. You see, this brother and this sister are exactly similar. So that is what you need to write. One thing is DNA, we need to identify DNA polymorphisms, first point. And second thing, we need to resolve those polymorphisms using DNA typing. That is the second point. And third point is you will have parents with diverse repeats will produce combinations of alleles. That's all you need to write to get all 10 points. 3 points. Parents with diverse alleles. This mom has 4, 6, this man has 1, 3. And when they produced 7 babies, only 2, some are similar, of course. This 1, 6, 1, 6 are similar. This brother and these 2 sisters also are similar. So you can just write, the first point is DNA polymorphisms exist in human populations. DNA typing resolves those polymorphisms. And the parents with diverse alleles, for example, 4, 6 and 1, 3, can produce a combination of children. That's all. And this is just one marker. You need to use 2 or 3 different markers in 4 and 6. Because if in one marker, say these 2 sister and brother are the same, you are doing 4 and 6, that one of these is a criminal. And then how do we identify if this person is criminal, and then this sister and this brother are finally the criminals for one this thing. And then you need to match, you need to create more DNA typing till you differentiate these 2 brothers, 2 siblings, to identify who is the exact sibling that left DNA sample at the crime site. Wonderful. So chromosome 17 is Holiday Junction. Holiday Junction, I think, is the previous chapter. Right? No, this one. Yeah. This is the Holiday Junction. So what is the formation and resolution of Holiday Junction? So at the crossover points, at the chiasma, or the crossover junctions, chromatids of DNA molecules of 2 chromatids come in contact, which is double strand breaks of chromatids make them into, create a junction of these DNA molecules. Like the blue is one chromatid and red is another chromatid. And then this is the point of chiasma where it formed a hybrid molecules, that is the blue and red combined, and then red and blue strand combined in the DNA. So this can resolve in any way. This can resolve this direction. If this is resolving in this direction, or it can resolve in this direction. In this direction, if it is resolving, you will see half of it is blue fragment and half of it is red fragment, like this. Or this orientation, site of crossover. So this one is, say capital A is here, capital B is here. So now this resolution alters capital A combined with B, small b, That is because of this orientation. Yes. Because here is the resolvase is acting here and resolvase is acting here. That creates a fragment that is capital A with small b, or small a with capital B, or alternatively this way. Well, both are same. This is capital A small a capital B, and here is also same. So it's just resolution, how it is resolved. So what we are going to write here is holiday junction forms at crossover, right? Where the physical point of chiasma that is bringing both chromatids together, that creates hybrid molecules of blue and red, or DNAs of diverse chromatids. And the resolvase that resolves longitudinally and horizontally creates crossover, which is capital A and small b, and small a and capital B. That's all you need to write. So question number 18. So that is question number 18 is chapter 5. So what is in question number 5? What is metacentric, submetacentric, acrocentric chromosomes? So metacentric is centromere is in the middle. Submetacentric is closer to one end of chromosome. Acrocentric is near end of the chromosome. Okay? Or acentric or dicentric. That's all. Very simple. Question number 18. Metacentric, submetacentric, acrocentric. That's all we need to write. Question number 19. Is this one. X chromosome inactivation. So the female mammals will receive two X chromosomes. For example, red and blue here in this figure. Or you can just write female mammals receive two X chromosomes. And then for few divisions, they all will be active. But soon in the descendant cells, one randomly gets inactivated. One in few cells, red is active. In few cells, blue is active. Okay, in somatic cells in the female. So how does this contribute to genetic mosaicism? This is how it is, right? And then provide examples. Here is one example, that is calico cat, where one X chromosome has black allele, and then another X chromosome has orange allele. So you have patches of orange and black. Also, you can write, because it is heterozygous for orange and black, one X chromosome has black, one X chromosome has orange. So either one is appearing, okay, in the patches. And also sweat glands in humans, in females. So sweat glands are also on the X chromosome. So some of the female tissues might profusely sweat, whereas males will have only two phenotypes, whether they can sweat or will not sweat. Okay, but females can be mosaic. Question number 20. significance of Y chromosome. So what is Y chromosome haplotyping? So a set of alleles of 2 or more loci present on the Y chromosome are Y chromosome haplotypes. Any chromosome, the alleles that are together of 2 or more loci are haplotypes. So using haplotypes, we can, we resolved a lot of human evolutionary histories, like Genghis Khan haplotype. Genghis Khan haplotype could be traced all over the Eastern Europe, where you have 10 of them, 15 of them, 7 of them, 8, 3. These are all descendants of Genghis Khan. Okay. But some of them do not have it. You go further down Asia, Genghis Khan never invaded these places. So they don't have any Genghis Khan haplotype people. Whereas in Afghanistan, there are 32 of them. In China, 6 of them. See, this is where Mongolia and all, definitely most of the people are descendants of Genghis Khan by chromosome. Kazakhstan, Uzbekistan, Afghanistan, probably Uzbekistan is here. This is Kazakhstan. All this are gypsies of India, are Bantha tribe, are the Jewish people, Jewish descendants of Bantha tribe of Zimbabwe. So there are 3 examples in the textbook. You can mention them to get 10 points. Discuss non-disjunction and provide examples of disorders. So what is non-disjunction? That's the process. So aneuploidy. So non-disjunction is failure of chromosomes to separate properly. Okay. One or other chromosomes makes to different poles and move to the opposite poles. So finally, when the chromosomes are split to the opposite poles, there are mistakes, there are errors. Either one chromosome is less or more. So this kind of non-disjunction is increased because some problems in spindle fiber formation and attaching it to the centromeres. That is because of there are chemicals in the environment, such as bisphenols. Okay. So what are the examples of disorders? What are the examples of disorders? Are euploid. Euploid is what? The balanced chromosome abnormality. So just like doubling of the chromosomes, diploid became tetraploids. Well, in plants, it can be tolerated, but not in animals. Or aneuploid. Aneuploid is one chromosome is extra, which is trisomy, or one chromosome is lesser, which is called typical aneuploid. Trisomy is also aneuploid. Oh, sorry, monosomic. Monosome means loss of single chromosome. Trisomy is addition of single chromosome, such as addition of single chromosome is chromosome 21. And polysomic is extra copies of single chromosomes. More copies. So examples are either monosomy or trisomy of most human chromosomes are unviable. whereas trisomies of 13, 18, and 21 are viable. Okay. So Down syndrome is chromosome 21. So that is about 21. How do deletions and duplications arise? Discuss the impact on gene function, referring the examples such as color blindness. This is already discussed. So the question number which question, folks? Copy number variation. Question number 11 and 12. Sorry, 22. 11 and 22, more or less similar answers. One is mechanism, and other is also mechanism asking in different way how the deletions and duplications occur. Because of unequal crossover, as well as pericentric inversions. Pericentric inversions or paracentric inversions, they can also cause deletions and duplications. And here it is asking again example as color blindness, which is nothing but unequal crossover. So deletions and duplications can occur in two ways. One is unequal crossover, and then inversion loops. So inversion loops can be paracentric and pericentric. If it is paracentric, that is centromere is outside that inversion loop, then there can be more deletions and duplications of the genes. Okay, so how the gene function, referring to the examples of color blindness. Color blindness results is completely because of unequal crossover, where two misaligned X chromosomes undergoing recombination and producing chromatids with various copies of various copy number variations for red and green. Okay. Chromosome 23, sorry, question number 23. Question number 23 is typically again same thing. The inversions. So this inversion, to match it, the normal chromosome will just bend, and then the inversion chromosome loops around. These are called what? Inversions, inversion loops. These inversion loops are two kinds: paracentric, where centromere is not included in the crossover. If the crossover is not happening, it is well and good. This inversion resolve in one way. This is normal chromosome, this is inversion. But if the crossover is really happening here, and then the centromere is outside that paracentric. Centromere is here. This is paracentric. So there you can have the dicentric chromosomes. Chromatids will have two centromeres, which will have huge problem of deletions. So if this kind of dicentric or acentric is there is no centromere. So these things are going to cause a lot of aberrations, abnormalities, because spindle fibers will pull this centromere to one end, this centromere to one end. In the tug, this gets broken, or this acentric fragment is lost. Okay, or if there is, now you see, centromere is within the loop. So this will not cause any breaks, but this will cause subtle changes. This D is now 3 copies, and A is only 1 copy. Another chromatid, A is 3 copies, D is just 1 copy. So pericentric inversions where centromere is included, more or less inversion loop is stabilized. That is the question. And Robertsonian translocation is this one. So if there is any acrocentric chromosome, like chromosome 14 and chromosome 21, are acrocentric or telocentric, where the centromere is on top of the chromosomes. So they can fuse and form a Robertsonian translocation. That is, chromosome 21 is attached to chromosome 14, and then in separation, this chromosome 21 goes to the one pole. And if this goes to the same pole, then the gamete will have 221 chromosomes, whereas the other gamete will not have any 21. Okay, so the gamete that has 221 chromosomes, if it is fertilized, then the zygote can be a Down syndrome zygote. Eventually, that baby will be Down syndrome person. So 3% of Down syndromes forms that way. So and then what is syntony? Syntony is this. Explain its importance. Well, syntony is to resolve ancestral genomes, the sharing of ancestral genomes. So the grass families, rice is the basal because it is the smallest genome, 400 megabase, and first sequenced everything. So the rice chromosomes can be traced across all the grasses, wheat, maize, foxtail millet, sugarcane, sorghum, and all these places. So for example, wheat one chromosome will have R5, R10, R5. So you don't have to write everything. You can just say grass families consists of rice, wheat, maize, foxtail millet, sugarcane, sorghum, all share the same genome that could be identified by sequencing the first rice chromosome. That is the smallest genome in the grass families. And rice chromosome fragments can be traced in the wheat, maize, foxtail millet, sugarcane, and sorghum. So using this information, we can create a hypothetical ancestral serial. Okay, if at all ancestor is there for all this grass, of course, that could have been there. That would look like this with all these domains, because all these domains are shuffled in different fashion to produce all these grass pieces. So that is question number 25. Let us move on, guys. And then what is molecular clock? We are going to chapter 14. Yes. This is chapter 14. So how do you explain what is the concept of molecular clock? What is the concept of molecular clock is fraction of sites that undergo change in some designated time interval. For example, number of replacement per site per billion years, especially in the housekeeping genes such as histones or globins. The change occur, fraction of sites will undergo only change, only fraction of them undergo change. So that can be compared. If an organism has 5 changes or 2 changes or 3 changes comparatively to another organism. So the one that has few changes is closer. The one that has more changes is further. And then it can differ dramatically from one protein to the other, of course. If you take one protein, you will estimate different clock. If you take another protein, that is different. And then, so this is the rough constancy in the rate of amino acid replacement over long period of evolutionary time. So the constant rate of change is called molecular clock. Provide some examples that follow this pattern is the globin genes and histones. They are there in the book, please. Especially, yeah. You can just write examples, globin gene and histones, that's all. That will work. Explain what is molecular clock and give the globin genes. And then what is the difference between species trees and the gene trees? Gene trees are not robust. That is based on the genes. Species trees are the whole genome level. So if there are species 1, 2, 3, 2 and 3 are closer to species 1, that is species tree, right? But a gene tree that tracks one particular allele, that is A1 became A2, and then that A2 split again A1 and A2. So finally in the species 1, A2 is fixed. In species 2, A2 is fixed. So when we see the species 1 and species 2 that has just A2 alleles, we cannot conclude that they belong to one species based on the gene tree, because gene tree is saying A2 are the common, which is not true. Though the species 2 and species 3, they have diverse alleles at that particular locus, A2 and A1. Because species trees are more robust, we can strongly depend on the species trees that shows species 2 and 3 are closer. That's all you are going to write. And 28 is understanding the synonymous substitutions and non-synonymous substitutions. So there are 60 codons, 1 stop codon and 3 stop codons. Synonymous substitutions are the nucleic acid changes that do not alter amino acid sequence. Okay, so the rate of substitution is high because they are not altering amino acid and they are uniform among many sites. So as long as, since they are not altering amino acid, synonymous substitutions are not purged out, they remain. Whereas non-synonymous substitutions that alter amino acid encoded protein and then affect the protein folding and then alter phenotype. So this kind of non-synonymous substitutions are lower. And then how do they affect function and adaptation? Well, non-synonymous substitutions create loss of function alleles. Okay, so that will affect the adaptation. Whereas in positive selection, some of the genes undergo non-synonymous substitutions that can produce proteins with enhanced function. So they are better, they are having very good adaptive role. So that is just 5 points, so you will write that. So please note, some questions are 5 points, some questions are 10 points. So the duplication, how it contributes. So first of all, how gene duplication contributes to evolutionary innovation. So first you need to understand what is an orthologous gene. That is the duplicated genes that are there in accompanied through speciation. So for example, human genes and cow genes or goat genes, globin genes, they are all orthologous genes. All the mammals have blood. So the globin is the one that is going to make hemoglobin. But the globin genes are different when you see at the sequence. But still they are sequence similarity because they have same ancestor. So they are all orthologous. Paralogous are within the genes, such as beta cluster and alpha cluster of globin genes in humans. Pseudogenes are duplicated genes that have lost the function. So question number 30 is Hardy-Weinberg principle. Hardy-Weinberg principle is, what is the ideal population? When the population is large, when the mating is random, there is no mutation and migration selection, then the gene frequencies and genotype frequencies remain constant. Okay. How do violations of these conditions lead to evolution? Of course, if there is any mutation occurred, then if that mutation is beneficial, that will get fixed, right? So that will cause shift from the Hardy-Weinberg principle. So the beneficial mutations will accumulate, contributing to the evolution. So question number 31 is inbreeding, allele frequencies and heterozygosity. So how the inbreeding works? So here what happens? The mating between first cousins in the pedigree charts are noted this way with a double line. And then the frequency of homozygotes increase when that kind of first mate cousins will happen. Homozygotes will increase. Whereas in the normal people, non-relatives, heterozygosity is below 2.5, 50% out of 1,000. Okay. Whereas homozygotes will really increase. So what is the impact of it? Because heterozygote mask the inbreeding, deleterious effects. Heterozygotes mask the deleterious effects. They are preserved, but matings happen between the relatives. What happens? Those deleterious alleles get exposed. Well, how inbreeding affects allele frequencies and heterozygosity? That is good enough for me. So question number 32 is describe the 4 evolutionary forces: mutation, migration, natural selection, and genetic drift. How do they impact, shape the genetic variation? Well, here mutation is the ultimate genetic source of genetic variation. Whatever mutations are happening, the neutral mutations will fixed and the beneficial alleles will get promoted. The migration is the movement of organisms among the subpopulations, dwindle the genetic structure of the subpopulations. Whereas natural selection, the process of adaptation, that is environmental perturbance, give rise to the disproportionate share of offsprings. The fittest offsprings will survive and then reproduce when they are exposed to natural selection. So the less favored individuals are eliminated. And random genetic drift is, a drift is happening such as bottlenecks, such as earthquakes or flooding or fire accident. Then the allele frequencies will change. And then especially in the smaller population is devastating. Then that kind of drift will affect the genetic diversity as a whole. So that is about it. Discuss the concept of heterozygote superiority. Heterozygote superiority is fitness of heterozygotes is greater than both individuals. That is especially in sickle cell anemia. The allele for mutant hemoglobin, globin gene, the 6th amino acid, is valine in place of glutamic acid. That creates sickle cell anemia, which is sickle cells. But in heterozygous conditions, that can provide some resistance to endemic malaria. being heterozygosity for sickle cell anemia is better because there is higher proportion of heterozygotes that are selected in the population. What can happen? When the heterozygotes mate, there will be 25% chance of producing a sickle cell anemia patient, which will die. That contributes to infant mortality. Because sickle cell anemia is in homozygous condition, it is fatal. Whereas homozygous dominant alleles will get killed by malaria. Okay, because they are very susceptible to malaria with normal blood. They find that kind of insects, mosquitoes find that kind of blood yummy, and then the malaria, plasmodium, especially parasite, will preferentially cause a lot of malaria in those patients, which are homozygous dominant. So heterozygotes prevalent. This is heterozygote superiority, a typical example. So how mitochondrial DNA is used to study the question number 34. Explain how mitochondrial DNA is used. So first thing is we need to discuss concept of maternal inheritance, where mitochondrial DNA shows maternal inheritance because the egg is the major contributor of cytoplasm in the zygote, and then mitochondrial genomes do not recombine. They can get mutated, but do not recombine. There is no crossover or recombination. So what is the out of Africa hypothesis? African populations have the highest level of mitochondrial diversity. That tells us mitochondria came from Africa. Clear proof. Because if you go and profile for African mitochondria, they have highest diversity comparatively to the rest of the world. And then you can match how those diverse mitochondria of Africa have kind of formed lineages outside. Within Africa, there are 4 lineages. African representatives are found in all lineages. So all these different mitochondria or mitotypes are spread across the Africa. Non-African contents will have only either of these lineages, Chinese, Indians, East Europeans, West Europeans, will have distinct type of mitochondrial DNA lineages that can be traced to Africa. So most recent common ancestor of humans lived about 170,000 years ago in Africa. So that is the answer. Then how the genetic drift impacts small populations differently from large populations. Discuss the implications for endangered species and conservative genetics. Yeah. Let us talk about this. You have 10 populations. 10 different flies are introduced in a flask to mate. Then their allele frequencies are here. So once the mating started from here, the populations were very diverse, but these are small number of populations, just 10. So what did it happen? It caused fixation of the populations. Some of the populations got fixed very early, and the diverse populations are there within that. They went longer to get fixed. So the frequency of alleles A is fixed. Capital allele, say, these populations have more capital A alleles. These populations have more small a alleles. And these populations are in between. And all of them are fixed at the 20th generation itself. So the drift happens because the genetic diversity is very low here. The drift can happen. So what is the implications of genetic drift in conservation genetics? Oh, a lot can be written, like we understood the dominant and recessive alleles. The dominant alleles get fixed so fast, and recessive alleles get fixed slowly. The dominant alleles getting fixed so fast in the sense the drift of alternative alleles, such as production of tuskless elephants in Zimbabwe. And then the allele frequencies like what we discussed just now using this Drosophila experiment. Okay, this is a experimental proof. If you are introducing small number of individuals in a flask and then forwarding them from one generation to the other generation for 20 generations, the populations get fixed either for capital A allele or small a allele. Small a is lesser. I mean, capital A is lesser. That means small a is more here. So in between. So all these populations got fixed so early. So as this one, you see, this got fixed. This population got fixed here. This population got fixed here, and this one here and here. So all the 10 populations, 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10, all of them are fixed. So here, some mating occurred, but since the population is less, the drift also happened quite fast. So that is about study guide, folks. I will send the study guide to you guys. Any questions you may ask now, or see you on Monday's class, tomorrow, where you will have quiz. And then on Wednesday, there will be test. So select your questions, make your notes, okay? Select your questions, make your notes, and prepare. Thank you all. Have a great afternoon. Take care.